Baruch ata Adonai. Baruch haba Yerushalayim.
Hello, dear students from G11. Um, how are you doing? This is our first video instructions that I'm recording. So, well, it's going to be probably really strange and terrible, but this is what we have, and then we're going to have to learn together how to do this. So, basically, I'm here just to explain how we are going to be on our activities during this next few or many weeks that are to come. And that will be based on basically three processes through activities that I'll be sending by clip. Also, we're going to have some book questions, so written material. The second way will be via video lessons that I will record. So this material will be probably uploaded on YouTube because I want to have some moments of expositive class. So probably we'll record so you can watch at any time. Another way will be by video conference. So we're gonna have some live moment that we can exchange, and we're gonna be exchanging whatever we can in these moments. Sometimes correcting exercises or making sure some points are understood. So that would be the three, let's say, pillars of our virtual contact. Okay, reading material, video lessons, and lives, right? For now, I'm just recording this as a test and also to give you the instructions for the next week. So, do you remember the work we've been working on before all this thing happened? We were doing this visual presentation of the protein synthesis and gene expression. So, you have to do a presentation with your group about the steps on the gene expression, and you have to present this in a creative and visual way by using materials and your creativity. So the idea will be kept, right? We're gonna do the same. We're gonna keep this work, but we're gonna work online. So virtually, you can produce this but not in a live presentation, will be recorded, edited, and in a movie or video. Uh, then you are free to choose the style. You don't need to show up your faces if you don't want. You can do like a stop motion movie. You can do a animation. You can do a PowerPoint presentation with someone talking behind. Uh, Whatever but you have to create this visual model of the steps of those processes we've been talking before. Um, the idea is the same, but you share tasks with the components of your groups. So one will be responsible for creating and designing, and the other one is responsible for actually recording the process, or different people recording different parts, and one person editing, putting it together, and then uploading to YouTube. I guess this is the best way. Um, it's easier for me to have access. It doesn't get too big, the file. But if you have any other idea, no problem. And also do a quick review of the process right? Just for refreshing about the content very quickly, and instructions. Okay, let's try. Thank you very much. So pay attention for the directions. Um, First, well, you're gonna have to keep the groups, but all the work will be done online, virtually. You don't need to get together with your group members, okay? Make sure you don't get together, stay home. Second, you have to produce a video or movie or animation, stop motion movie or PowerPoint presentation. Be creative, right? Third, you have to share tasks. That's very important to share tasks between the members of your group. So each one will be responsible for doing one section, like transcription. Another one is responsible for translation. Someone else is responsible for compilating all together. Someone else for edition. If you want to have a narrator, someone can do that, or someone responsible for the text information add to the video, right? Communication within the group, it's a very important thing. So it must be organized in a way that you can do this division. Once complete, the project must be uploaded to a platform. I suggest YouTube, but if you have any other idea, uh, and the link must be sent by email to me. Okay. Evaluation, basically three components: the content itself, the communication skills, so the way it will be presented, if it's clear, if it's good to see, if it can be totally understood in your presentation. This counts as communication. And also, of course, creativity. It must be entertaining somehow. That's the idea. Your sources will be, well, anything in internet about. I suggest always BioNinja, which is the website I'm using during my classes. Is quite complete. And also your book have a lot of information too. But internet generally has a lot of information about this subject. Well, now a quick review about what we're doing. So we were doing this, we were looking at this process called gene transcription, right? Which is the first stage. So gene transcription is just to refresh uh, your mind. It's this process. We already had this class, I know, but let's talk about this very briefly. So the process of gene transcription is the first process of the protein synthesis. So it starts basically with a RNA polymerases, which is an enzyme. You have to somehow create this enzyme. Uh, and you have as base one of these strands of the DNA, remember? Uh, and the product will be then the RNA strand produced. So this is considered transcription, a production of RNA out of a DNA piece. Then you have to remember this coding thing, right? So you have to remember that in RNA, you don't have certain nucleotides like thymine, for example. It's only present in the DNA molecule. So in RNA, the substitution of thymine will be done by another nucleotide called uracil. So how you recognize an RNA, basically a single strand, and also the presence of uracil. Then you make sure your RNA. You have a right direction, remember? So the polymerase produces an RNA which is 5 prime up to 3 prime and runs on top of a DNA strand 3 to 5 prime. Okay, it's quite confusing, so pay attention in this direction of the transcription, right? I gave you the gene array sequence, so you have to work on that piece of sequence that I provide in the class. If you don't have the sequence, well, you're gonna have to talk with your friends, because I remember some people took pictures of it. Then we have the genetic code, that is not something you need to learn really. It's just something that you need to check. A table, codon. Okay, a codon is a sequence of 3 nucleotides, and they will codify for one amino acid. Okay, out of our table. So every 3 nucleotides in mRNA, we are going to have one corresponding amino acid, right? And they will be placed together by the ribosome, remember? So you need to provide the ribosome structure as well. This will generate a polypeptide, so you promote these peptide bonds, but according to this table. This table is not the best table, but it can be used. You also have a table in your book if you want, if you prefer, which is in a simple way to understand. Piece of cake, right? Then you finally have the process called the translation itself, which is when you translate the nucleic acid, which is the mRNA, into a polypeptide protein. Then this is called translation. So the transcription is in the same language, let's say, is nucleic acid, which is DNA, to RNA. The translation then is another language, is nucleic acid to polypeptide or proteins. When I see your polypeptide, I must be able to go back and find the same gene that I provide you. So I want to see everybody producing the same polypeptide at the end. They can be very different in the way they look, but their code, their sequence must be the same to be correct. Okay. Uh, well, that was a very fast review, and all the information you need is in your book. Internet is plenty of information, so if you have any other further questions and has to be solved before the project, then email me. Thank you very much, and I see you soon.